So good morning, everybody, and thank you for this invitation to give a talk about the microbiome and cardiometabolic risk in HIV and non-HIV populations. I have no conflicts of interest. So I'll start with sharing some data on the microbiome and cardiometabolic risk in the general population, and then move to HIV populations and discuss if HIV is any different. I'll also try to discuss where we are today and what should be the next steps in research. So there is a very complex interaction between diet, the microbiome, and cardiometabolic risk. And I've tried to sum up the most important topics on this cartoon. And clearly, if you read the literature, the most attention has been to this metabolite, TMAO, which is a potential link between westernized food, the gut microbiome, and cardiovascular risk. So in order to produce this metabolite, you need 3 steps. First of all, you need to eat certain nutrients, such as carnitine, which you find in red meat, orphosphatidylcholine, which you find in eggs, in dairy products, and in certain kinds of fish. But vegans do not produce this metabolite. Second, you need to have an intact gut microbiota. 1 week of antibiotics is sufficient to suppress the production. And finally, there is an oxidation step in the liver from trimethylamine to the oxidized form, which has been shown to produce atherosclerosis in animal models, but not if you add antibiotics to the drinking water, really suggesting that this is caused by the microbiome. So in humans, TMAO can be used as a biomarker, and it strongly predicts myocardial infarction, stroke, and death, as shown several times by the Stanley Hazen group. We looked at this in heart failure several years ago, finding that in the upper tertile of TMAO, only 50% of the patients were alive 5 years into the study. So it's a really strong signal. And more recently, the Stanley Hazen group has also looked at TMAO in a more acute setting, and elevated levels of TMAO also predict new cardiovascular events 30 days after a myocardial infarction. And whether TMAO is ready for use in the emergency room is still a question, I think. It's interesting that it seems to interfere with platelet reactivity, but so far we have enough established biomarkers for clinical use, but it's still a very exciting biomarker. But what about HIV populations? Is TMAO a potential biomarker for clinical use? A few years back, we measured TMAO in almost 1,000 plasma samples in a nested case control study of HIV-infected individuals with or without myocardial infarction. We have followed these patients for several years, from before starting ART to the last sample before the myocardial infarction. And we find no signal whatsoever of TMAO, so it's not useful as a biomarker. What we do find is an increase in TMAO levels after start of ART, and we find the largest increase in those starting protease inhibitors. So we speculate that protease inhibitors might interfere with the liver stage. It's known to induce a lot of enzymes in the liver, making TMAO a less useful biomarker in HIV. So the second metabolite that has really been emphasized in literature is butyrate and also other short-chain fatty acids. So butyrate is a fermentation product from dietary fibers. It has local anti-inflammatory effects in the gut, and it's also the main substrate for colonocytes. So if you have a decrease in butyrate, you tend to have increased permeability in the gut and increased leakage of LPS and other products from the microbiota. We got interested in butyrate after performing this study in heart failure patients, where we have reported microbiota profiles from 2 independent cohorts, and we only report the alterations that we see present in both cohorts. And what is striking here is that almost all the microbes that are depleted in heart failure belong to Clostridia, and they belong to the same bacterial families. It's Lactospiraceae and Ruminococcaceae. And these are known to produce short-chain fatty acids such as butyrates. We also see that the overall genetic potential for producing butyrate is decreased in heart failure. And we even see that some of the microbes that produce butyrate seem to be lowest in those progressing to cardiac transplant or death. So potentially linking this to prognosis. And if you look at the contemporary studies of microbiota in cardiovascular disease, there is one common finding. It's a reduction in different Clostridia, such as Roseburia, Fecalibacterium, Eubacterium, Ruminococcaceae, Lactospiraceae. The common trait here is that they have the potential to produce short-chain fatty acids. The weakness with these studies is most of them are small. It's only 16S, not metagenomic sequencing, and most of them are not adjusted for confounders. We found in our cohort that most of the dysbiosis associated with heart failure was also linked to low dietary intake of fiber. So diet, food frequency questionnaires are necessary to adjust for in these kinds of studies. So how about the HIV-related microbiome? If you read the published studies from 2012 to 2016, I think there are some common traits. The studies report differently when it comes to biodiversity. A lot of studies finding lower diversity in HIV, but also some finding increased diversity. But the common findings is, as most of you will know, decrease in Butyrates and increase in Prevotella, which we for many years thought was the HIV-related microbiome. But then this study came, which really changed the field completely. So in this study, gut microbiota is linked more to sexual preference than to HIV infection. So the individuals here, they are sorted according to a prevalence of Butyrates, which is associated with non-MSM. Prevotella is associated with MSM, men having sex with men, rather than HIV status. So after this, we had to redesign the studies. This is the Copenhagen-Oslo HIV Comorbidity and Microbiota Study comics, which was recently published in Clinical Infectious Diseases. Here we have tried to define an HIV-related gut microbiota independent of sexual preference and other relevant confounders. And second, to relate the HIV-related gut microbiota to excess cardiometabolic comorbidities. So in the comics study, we have applied 16S Illumina sequencing on 405 HIV-infected individuals who have been stratified for MSM and non-MSM status. We also analyzed 111 controls, both from the general population and also MSM from a PrEP cohort. And the first finding from this study is that MSM and HIV seem to have a opposing effect on the microbiota diversity. You see here that the groups with MSM, they tend to have a higher biodiversity, whereas the groups with HIV seems to have a lower diversity. So this might be one of the explanations for the opposing studies published previously. Next, we wanted to look at the HIV and MSM related microbiota changes by comparing different groups. So here we have used a program called Metacoder, where we build phylogenetic trees and we compare 2 and 2 groups. So for example, here we have compared an infected MSM with HIV MSM. And here we have compared the general population with HIV non-MSM. And we propose that the differences we see both in this panel and this panel is linked to HIV rather than MSM. So what are these changes? First, we see an increase in gamma proteobacteria and the sulfovibronacea, which both belong to proteobacteria. These are typical LPS producers. And second, we see a reduction in a lot of Clostridia, a lot of these short-chain fatty acid producing Clostridia. So this is what we think or find to be the HIV-related microbiome. And reassuringly, when looking at literature, this is a very nice review by Vujkovic-Sivin, sorting the previous studies according to whether or not they are adjusted for MSM status. And those studies which are adjusted for MSM status really report the same findings as we do in our study. Increase in proteobacteria, increase in enterobacteriacea, and increase in the sulfovibronacea. And also a reduction in these typical short-chain fatty acid producing Clostridia. So next question is, are these changes associated with the metabolic syndrome? First of all, most of the microbes that are increased in HIV are potential LPS producers. The sulfovibronacea is also a potential producer of hydrogen disulfide, which is a known metabolite that could be toxic to the gut. So this could be linked to microbial translocation and gut damage. And we see that this trait is associated with the metabolic syndrome, twofold increased risk for metabolic syndrome. And this risk is really driven mostly by the diabetes component, threefold increased risk, and also increased risk for elevated triglycerides and possibly also hypertension. So we discussed when writing this paper, is this microbiota specific for HIV, or is it more a general trait that is linked to obesity? When we wrote this paper, we saw a contemporary paper in Science that was published last summer, showing some of the same traits related to obesity. They find that outgrowth of the sulfovibrio at the expense of Clostridia is sufficient to drive obesity in immunodeficient mice, and potentially because of increased lipid absorption from the gut. And they propose that T-cell mediated regulation of the microbiota is a protective shield against obesity. So if this is the case, is it possible to find an interaction between immunodeficiency and microbiome in causing metabolic syndrome in HIV? So all of our subjects, or most of them, are on ART, but we have stratified for nadir CD4 counts. And we see here that the odds ratios are really increasing in those with a very low nadir CD4 count. The confidence interval is obviously very broad, but there seems to be an interacting effect. We have also looked at adipose tissue measured by CT scan. And we here also see the same interaction between immunodeficiency and the microbiome. So the HIV-related microbiome was associated with 30 square centimeters larger area of visceral adipose tissue in those with severe immunodeficiency, but not in immunocompetent patients. So finally, the last topic is gut permeability and microbial translocation, which is reported in several cohorts of heart failure and diabetes. And as most of you know, also is a topic in HIV. After this seminal study by Jason Brenchley and Danny Dueck, showing that microbial translocation is a cause of systemic immune activation in chronic HIV infection. And this study inspired a lot of work also from our own group. So in several studies, we have measured LPS, and we have found it to predict hypertension, to associate with dyslipidemia, insulin resistant, Framingham score, and also trunk fat and the metabolic syndrome. However, no studies, as I know about, have shown that LPS really predicts hard clinical endpoints. CD14 does, but this is caused probably by activation of the toll-like receptor for complex and inflammation. So could there be differences between different forms of LPS? In this paper, we have looked at hexa and penta-acylated LPS-producing bacteria. So hexa-acylated LPS is known to trigger inflammation on the toll-like receptor 4 complex, whereas penta-acylated LPS is probably more inert. And this is a probiotic study from HIV, where we see that after probiotic, the abundance of penta-acylated bacteria decrease significantly. We see the same numeric decrease in hexa-acylated bacteria, but this is by far significant. And this is perhaps because penta-acylated LPS outnumber the hexa-acylated LPS-producing bacteria with a factor of 25. And we also saw that changes in plasma LPS correlated with changes in gram-negative bacteria, which is almost entirely driven by changes in penta-acylated LPS. So from this study, we conclude that a lot of the LPS that you measure in plasma is perhaps triggered or released from penta-acylated LPS-producing bacteria, and it's perhaps not linked that much to inflammation. So finally, I'll show another way of looking at microbial translocation by looking at extracellular vesicles. So this is a recently submitted work by Beate Westad, who has looked at extracellular vesicles. She's able to detect them by electron microscopy in cohorts with HIV, with and without type 2 diabetes. You see them here in red arrows. There's a strong correlation between concentration of these EV particles and LPS measured in plasma. And she has also been able to detect 30 proteins from bacterial sources, most of them belonging to proteobacteria, as we know is part of the HIV-related microbiome. And when doing proteomics, we also see enrichment of IL-6 and IL-1 beta in subjects with HIV and type 2 diabetes. And we have recently shown that soluble markers of interleukin-1 activation predict first-time myocardial infarction in HIV-infected individuals. They have approximately 50% increased risk of first-time myocardial infarction when this system is activated. And interestingly, we see some of the same signals in COVID-infected patients, possibly because of LPS priming the inflammasome. So to conclude, the microbiome of HIV and cardiometabolic diseases have some common traits, including depletion of Clostridia and impaired gut barrier. But there are also obvious differences, for example, TMAO being strongly predictive in the general population, but not in HIV. Major confounders include sexual preference, diet, and medication. And in order to move the field, there's a need now for well-designed large cohorts analyzed by metagenomic shotgun sequencing and also exploration of microbe-host interactions before planning targeted interventions. So finally, thanks for my co-workers at Rikshospitalet, led by Susanne Dam Nielsen, and my co-workers in Oslo. And thanks for your attention.